Good evening and welcome to Cook the Science. My name is Thomas Michaels. I'm a professor of soft and living matter physics here at the Department of Biology at ETH Zurich, where I'm also a member of the Bringing Materials to Life Consortium and also now of the World Food System Center. And it's amazing to see so many of you here tonight for what is actually the last Cook the Science lecture of this academic year. I find it amazing how time flies, but we are already through one year, and I think it has been an amazing experience. And if you have been also to the previous ones, I hope you enjoyed. And now with Cook the Science, the idea is very simple. What we want to do is we want to bring science closer to the public, closer to you, in a way that is relatable and enjoyable. And what better to do this than something we all love, which is cooking. And so at each event, we have a topic from the kitchen science department, and we try to make sense of it, both from a scientific and chef perspective. And we have also the help of chefs and scientists that help us to really understand the two elements. And the topic tonight is indeed about cooking, but perhaps a little bit of a weird or a different way of cooking, but that maybe perhaps not many of you are familiar with, which is fermentation. And we will actually focus on one specific type of fermentation, lactic acid fermentation. And to tell us more about this fantastic phenomenon, we have two amazing guests here tonight. Patrick Marxer from Das Purer, so owner of Das Purer in Wetzikon. From in the neighborhood of Zurich. And also we have Markus Schuppler from ETH, so from the food microbiology laboratory here at ETH, and he's really an expert in microbiology and particularly also with what concerns aspects of food safety that we will discuss when making fermentation. And this is a really wonderful pair because I've actually heard that you have had a collaboration between Das Purer and ETH for over 10 years now. And so tell us a little bit more about this collaboration, Markus.
Yes, first of all, thank you to be here. I'm really glad about that. And yeah, we know then more than 10 years, I would say. And actually, I even know Patrick without his bowler hat. Imagine.
And without the beard.
And I'm really happy that we could establish collaboration during the last years. And it's a collaboration where Das Purer acts as a practice partner in the student course Agri-Food Projects. And in this student course, mixed teams of agricultural students and food science students work together on real problems and questions of small and medium enterprises or farms as practice partner and create a win-win situation for all involved parties. What you can see here on these images is a former project where the students worked on the improvement of the shelf life of fermented products from Das Purer by transferring the results and findings from the lab later then to the production unit of Das Purer. And at the end of the student course, they provide their results and findings in a presentation.
Fantastic. So really two experts on fermentation. And I would say we have really a lot to cover. It's a complex topic. And so I think we should get started right away.
Okay, now it's my turn. Okay, good evening, everybody. Who has dranken or eaten something fermented today? Come on. Who has had a coffee today? Who has had a tea today? Who has had a dairy product? Who has had, I don't know. Go ahead, Markus. All these things are fermented food. All of these things have something to do with lactic acid. So please, we are always using fermented stuff as food in any way, and a lot of things we don't even notice that we've used it. Go ahead. This is not something absolutely modern. We're trying to make it modern in the moment. Everybody's lactic acid, or I've heard, I've had enough kimchi everywhere and things like that. I understand that very well, but no, the whole fermentation thing is about 10,000 years before Christus, BC. It started, and it was mainly in the Asian part, the Near East, or in Egypt, where we had fermentations. And these are a couple of slides which show how milk, dairy mainly, was fermented. Then we have typical fermentations. Like I said, kimchi is something more east. Then in Africa, there is a Milton fermentation, which is made out of milk, which is used often. And here in Europe, it's sauerkraut, which is, something very typical, but also dairy products like yogurt and things like that, which have been used very early already. The absolute start of fermentation as a topic in science was around 1850 in the 19th century due to Mr. Pasteur, which really made a big, huge thing out of that. Thomas, please go ahead.
Yes, I mean, this is really interesting because, if you think about it, we have been fermenting for millennia, yet our scientific understanding of fermentation is relatively new in this context. But of course, this can help us a lot nowadays to make even better products. And so I think this scientific understanding coincided also with the developments from the side of the instruments. And in particular, it ties very relatedly with the history of microscopy. And so this actually dates back to Hans and Zacharias Janssen, so father and son, who at the end of the 1500s developed the first compound microscopes. And then this development then, of course, made their way into various disciplines, including biology. And then it's because of these instruments that then people like Robert Hooke were then able in 1665 to observe things like cells. So he was the first person to coin the word cell. So this is actually the microscope that Robert Hooke used, and this is a drawing of what is so under the microscope from a sample from an oak tree, and what you can recognize here are cells. And an interesting fact that I actually didn't know is that the word cell, he came up with this word because these subdivisions here that he saw under the microscope reminded him of rooms in a monastery, so like the monks. And so these are cellular, and so that's where the word cell actually came from. And then the history goes on with other people, so Antoine von Leeuwenhoek, who was the first person to describe bacteria, also observed under the microscope, so one of the fathers of microbiology. And then, of course, to people like Louis Pasteur, so who, as Patrick has said, has really made a huge contribution to our understanding of fermentation. So this is actually the microscope that Pasteur used. And so what did he use this microscope for? Well, so he used this to understand the process of fermentation. Historically, the first one, excellent. We will have quite a few equations, so I hope you are ready to clap. So this is the example of alcoholic fermentation. So it's the first one that was described where glucose is transformed into ethanol. And what he realized is that actually this process is due to the action of very, very tiny organisms. So in this case, it's very, very tiny yeast. And it's really due to the action of this yeast that this process goes. And so this was really a fundamental shift in our understanding of fermentation. And another very interesting fact is that the word fermentation comes from the Latin verb fervere, which means to boil. And in fact, the ancient Romans, they used this term to describe what happens when they took grapes and they made wine out of it, and they saw that these grapes were actually boiling, were forming bubbles, and they thought this is like boiling, so they used this word. But of course, now we know that this is not boiling in the strictest sense. It's actually the action, the work of this yeast that eat the sugar, produce ethanol, and also produce CO2, this gas, in this process. So really, this is changing the way we think about this process. But this now is only one example, which brings us to what is, we'd say, a bit of the general definition of fermentation, and at least a useful one for what we will do tonight, which is, in general, fermentation is the transformation of food by the action of microorganisms. And to be more precise, this process happens in the absence of oxygen. And so apologies for my understanding of fermentation. I'm a physicist, so this is how a physicist thinks about fermentation. So you have a microorganism, which is almost like a sphere. I've made it a little bit more complex for obvious reasons. And so this microorganism eats different complex molecules like sugars and proteins, and it transforms these molecules through biochemical reactions into energy. So the overall goal is to produce energy, and this energy is then used by the microorganism to then thrive, to divide. And then through this process, it also produces a set of byproducts, and which byproducts they are depends on the type of fermentation, and they include a really wide range of compounds from alcohol, also acids, very important, so lactic acid for our case tonight, also gas, ammonia, and all sorts of molecules that we like because they give flavor to our food, and they make the food really delicious. And I hope also tonight, if you have not really had experience with fermentation, you will experience all these beautiful flavors that are created by this microorganism. But now, of course, through the development of our understanding of biochemistry, we now know actually a bit more of how these biochemical transformations work. And we can actually update a little bit the definition here by saying that this transformation is due actually by enzymes that are produced by microorganisms. So these enzymes are molecules that facilitate, accelerate specific biochemical reactions. And if you are not familiar with the world of enzymes, a very little trick for you to remember them, they all end with the A's. So you will actually recognize them later on in the talk. So now to go back to cooking. So I said fermentation is like cooking. Actually, indeed it is, because it's all about allowing microorganisms to break down complex food into simpler, more easily digestible and delicious molecules. And so in this sense is actually really a form of cooking, although this might seem a little bit weird because we are more used to the idea of cooking by applying heat, okay? But actually there are quite a few resemblances between cooking with heat and cooking with microorganisms, tonight lactic acid bacteria. And it's the fact that in both cases, what we are doing is we are actually breaking down these complex, larger molecules into smaller compounds, which have more flavor. So in general, these compounds, smaller molecules, are more easily detected by our tongues, which is why fermented food is actually so delicious. And no chef on the planet, I would say, can do to food what bacteria do to food. It's amazing. It's really something amazing, and we really take advantage of this. So it's a really fascinating area that I'm also been discovering with Patrick and Markus. So now, the topic tonight, as I said, is lactic acid fermentation. So I will give a very brief introduction to this, again, from a physics perspective, and then Markus will correct all my mistakes later on. And so lactic acid fermentation is due to the action of lactic acid bacteria. So the microorganisms tonight, these are our friends, lactic acid bacteria, and they are found on the skin of fruit and vegetables. So they are here. They are also on our skins. So they are right there, ready to be used. And we will actually observe some of these bacteria later in the microscope that we have here, that Markus brought. And we also try to make some drawings of how these different bacteria look like. So Markus, can you explain a little bit so that we can actually recognize them later on in the microscope?
Yeah, what we see here is we have two major morphologies of bacteria. We have the round spheres, or more or less round spheres, which are called cocci. And sometimes these cocci, they form chains. So we have streptococci. I hope we will see them later on also under the microscope. And of course, there's lactobacilli. So bacilli means we have these rods. And these two is the major types that we find then also in the fermentation.
Yes, fantastic. So look out for these guys. Now, very briefly, the reason why we like lactic acid bacteria is because there is a formula. is because they use sugars that are in the vegetables and in the fruits, and they produce lactic acid in the absence of oxygen. So this is really the acid that then gives the sour flavor to things like kimchi, like Patrick has said. So we will look at it later on. And in many lactic acid fermentations. Now, I'm going to mention a few important facts about lactic acid bacteria. Maybe they won't make much sense to you now, but I will explain. So first of all, because these lactic acid bacteria produce lactic acid, they actually are able to tolerate, they are happy in an acidic environment, which means low pH. And also another important aspect is that unlike other, especially pathogenic bacteria, they can tolerate relatively small amounts of salt. And this is why we add salt when we make our preparations. So why am I saying this? It's because the job of the fermenter, it's really about knowing which bacteria you want to look at. So in this case, our friends are the lactic acid bacteria, and you want to make sure that they thrive, that they are happy in their environment such that they can do their job, which is to transform our food, make it delicious, produce all these beautiful compounds that we love. And so they have to be happy. And at the same time, we want to make sure that pathogenic, bad bacteria are taken away and they don't thrive. And so these conditions here are very important to actually ensure that we have the right conditions for the lactic acid bacteria to thrive, and at the same time to make sure that the pathogenic ones are kept away. And actually, when we were preparing the talk, we had this discussion, and Patrick, you came up with this picture of, analogy of like a party.
Exactly. We were talking about how does this work with these bacteria, and I explained to Thomas, it's like a party where everybody is invited to come in. And then the DJ starts to play music like techno, and then half of the people leave because it's not their music. And then somebody says, all those who don't drink alcohol, who drink alcohol have to leave also. Okay, so they go also. And at the end they say, those people with pants leave also. So those are also out, and then you only have the specific persons in the party which want exactly this situation.
Exactly. And so as we will see, these pathogenic bacteria are already present. So we really want to make sure that they are left out of the party. And so the point is we want to understand, control the conditions such that these bacteria can thrive and the other ones don't. And so we have to understand a little bit how these bacteria thrive in their specific environment. And so we know that bacteria multiply very fast. They do that exponentially. Because if we start with one bacterium and then this divides after a characteristic time tau, which is the characteristic multiplication time of the bacterium, we have 2 bacteria, and then after another time tau, we have 4 bacteria, and then after another time, we have 8 bacteria, and so on and so forth. So you can see how these bacteria multiply very fast. And so in general, after n steps...
Got one for free, huh?
Yes, exactly. Very good, very good. So after n generations, actually, we have 2 to the n bacteria. And so the number of generations n is actually t divided by tau, where t is the current time and tau is the multiplication time. So if you want, is the rate at which these bacteria multiply. And what is really important to understand is that this multiplication time depends on the environment, depends on the temperature, depends on the pH, depends on salt conditions, depends on oxygen, and all sorts of conditions that we can actually create for our bacteria. And this is really important. This is why salt is important, and so on and so forth. Now, to give you an idea of how fast this multiplication actually is, let me make one example of E. coli, which is one of the fastest growing bacteria. So at 37 degrees under optimal lab conditions, it multiplies every 20 minutes. So the question is, after 1 hour, how many bacteria do we have? Well, it's 2 to the 60 divided by 20, so it's 8. And then after 15 hours, we have actually quite a big number. Look at this. So this is 3.5, please clap, yes. So 3.5, 10 to the 13, which is actually 4,000 times the whole population of humans on Earth, just in 15 hours. And then 20 minutes later is already double that. So it's incredible. And to give you a better understanding, like the mass of one bacterium is about 10 to the minus 10 grams. So this number of bacteria would make 3.5 kilograms of E. coli, which is quite a bit. So really, we get very fast multiplication and a lot of bacteria. But so is this actually realistic? I mean, do we get really these huge numbers? Well, of course, bacteria multiply exponentially. This is true, but up to a certain extent. Thank you. So this is the formula for how exponential growth works. So the rate of change of the population depends on the current population with the growth rate kappa. But of course, this exponential growth cannot go on forever. At some point, we run into resource limitations. So there are finite resources. And therefore, the population at some point stops to grow because we run out of these resources. And so the equation now it gets modified by this term here with the L, which is the resource limit. So this is really the resources that are available in the environment, and this is known as the logistic growth equation. And so this typically describes the growth of bacteria. Now, this is only one species of bacteria. But of course, another very important element that controls the growth of this bacteria is competition. Yeah, I told you we would have quite a few equations tonight, and they're getting more complicated. So the idea really is that actually we have many types of microorganisms, and they all compete for the same resources. And so this is described by this equation here, where Ni is the number of microbes of species I, and then kappa I is the growth rate of species I, and then we have an interaction coefficient alpha ij, which describes the interaction between species I and species j. And so this then gives rise to these complicated, complex time courses of the population, where you see how different bacteria or different types thrive at different times. So initially we have one type that then goes away in favor of a new type, which then dies out, and then another one kicks in. And so you see really how we have this competition between these different species. So it's really a complex environment that we have to control and to make sure that only the right ones are thriving. And so to do this, actually the fermenter has quite a few parameters at disposal, and in particular temperature and in particular salt is very important for this. And so I think I've spoken enough, and you have had enough equations that I think we need a break. So we should start with the first fermentation of tonight, which is a dry lactic acid fermentation. And so you find actually the recipe here in the QR code. Here, you can download it from the website, and the floor is yours.
Okay, great. Where's my cameraman, please? Christian and Lily, you can open the box. We have, just to explain, we have prepared a fermentation, which often is terrible to smell. The smell will go away in about 20 minutes, so...
We called it the bomb.
Yeah, we called it the bomb, exactly. And Markus will then pleasantly explain in 20 minutes or 30 or 40 minutes what happened there. Okay, so we're making a dry fermentation with asparagus. That's the green asparagus you have on your plate. You can try that. It's a dry fermentation. We made it 13 days ago, so it's pretty old already. What I have is I've peeled white asparagus, which I put into a vacuum bag. And then 2% salt. We have here 500 grams of asparagus, and very important, the salt. We'll come to that later on. It has to be non-DI'd, right?
Non-iodized.
Iodized, thank you. Very important because the iod is really something that kills your bacteria. So 2 grams on on 100 grams, I have 500 grams. So that means I'm just going to put 10 grams on into the bag. And it seems complicated, but it's finished. That's it. We're going to seal it, and that's it. Of course, you can use some ginger. You can put some kind of spices into it. It really doesn't matter. But the product you have now eaten, the green asparagus, we only put a little bit of estragon into it, and that's it. Nothing else, really. So the whole thing is now seal it with some kind of a vacuum sealer. These private ones work. Also, let's see if this one works. It's not mine, so I'm not responsible for it.
What a lovely sound. Yes, and also we will come back to the point of why we do it in a vacuum-sealed bag, because of the oxygen, and Markus will explain very nicely why this is very important as well.
Okay. So that's the first presentation. And what will happen now, in the next couple of days, it will grow as a balloon.
So how long do you keep it?
Oh, you can keep it. After 5-6 days, it already works. But as I said, these asparagus are 13 days old. And because you don't have more sugar than it has, you won't have it. It won't go further on.
Yes, and you keep it at room temperature.
Exactly, yes.
Yes, very good. And then
if you want to stop it,
No.
If you want to stop it, then you can cool it. Asparagus is not a problem because it doesn't have that much sugar, so it won't get too high in the acidity. Like sauerkraut or something like that, if you leave it a couple of months, you'll get a terrible acidity, which really, I don't like it that way. We like it really pleasant. Our acidity has to give it to taste. It doesn't have to kill you.
Fantastic. So now let's go back to a little bit of science, and let's try to break down some aspects of the recipe that we've just seen. So I've told you about the importance of creating the conditions such that the bacteria are growing fine and the pathogens are not growing. And so one element, really, we had just a few ingredients, and one of the few ingredients was salt. So why do we have salt? So what is the effect of salt? And so very loosely, again, as a physicist, salt, you probably know, it also has antimicrobial effects. And the reason why it does that is because it can kill bacteria. And the way it does this is by using physics. So it's mostly through osmosis, which is effectively like the movement of water when you have different concentrations, for example, of your salt. And so the idea is that if you have an environment where the salt concentration between the inside and the outside doesn't balance, then you will have an osmotic pressure, which then draws water out of your system, and that actually can have consequences. And so here, pi is the osmotic pressure, and delta C is the salt concentration difference. Now, of course, these high concentrations of salt are bad, but as I said, mild concentrations of salt can be tolerated by certain bacteria. And here we are lucky because lactic acid bacteria are so-called gram-positive, which means that they are somewhat a little bit reinforced in their wall because of this peptidoglycan wall here, which actually means that they can actually tolerate some level of salt, about up to 10% of sodium chloride, which is table salt that you have at home, non-iodized. And so this is actually very important because not all bacteria tolerate this amount, and in particular, as we will see later, the pathogenic bacteria don't tolerate these salt concentrations. And so typically for the fermentations, we use 1 to 3% of salt as our insurance to keep the bad bacteria away. So how much percentage of salt did you use in your fermentation? 2. Yes. So T2 is really the typical number, and it's really important that you have enough salt to create these conditions. And then you also mentioned an interesting aspect that I actually didn't know, and this is really an important trick, about salt with or without iodine, because you know salt is iodized typically when you buy it in the supermarket. So Markus, can you tell us a little bit more about why this is the case?
The answer is very easy, because salt is, oops.
It's not that easy.
The sun, yeah, that's very Because iodine is healthy for us, but not for the bacteria. I mean, you know that from wound treatment, usually if you have wound treatment, very often they contain iodine. So this is the reason behind that. And now I wanted to say I deserve now a very huge applause because my next slide this is all equations, chemical equations. But why do I show you this slide? First of all, you have to be aware and pay attention now because this is part of the final exam at the end of this lecture. So be aware of that. And the second reason is we heard that fermentation, the procedure is rather easy. It's easy, it's simple, just take some vegetables, add some salt, and that's it. But this is what is behind the scene. So this slide shows you the whole complex metabolic processes that are driven by the lactic acid bacteria in your fermentation. This is what happens. And Thomas already said we have many enzymes involved. So the bacteria, they produce these hydrolases, for example, to break down the carbohydrates. We have proteases that break down the proteins, further peptidases that produce then amino acids from these peptides. And then these products here, they are used then by the bacteria to gain energy or to produce biomass. Yeah? And what is very important to understand what happens in lacto-fermentation or lactic acid fermentation is that within the lactic acid bacteria, we have two different groups. And this is the two groups. We have one group, it's called the homofermentative lactic acid bacteria. And what they do is they produce two molecules of lactic acid from one molecule of glucose. And then we have the other group. The other group is the heterofermentative lactic acid bacteria, and they produce only one lactic acid molecule, but they produce CO2 and they produce ethanol, and some of them also produce acetic acid. So we have these two groups, and both of these groups play a role then in the fermentation. I want to show you an example for these groups. For the heterofermentative lactic acid bacteria, this is leuconostoc. Again, we have this cocci that form a chain. So this is typical for the leuconostoc. I hope we can see that later on then under the microscope also. And then we have example for the homofermentative lactic acid bacteria. This is some species of lactobacillus and also lactococcus lactis. You can see here again the cocci and the bacilli here on this slide. They are typical homofermentative bacteria. Okay, if we start a fermentation, what happens then? Thomas already said to you that the bacteria are already on the vegetable, so we don't have to add a starter culture. So we call this a wild fermentation also because the bacteria are already there. But what you can see here, in the beginning, you have a lot of other bacteria and only a few of lactic acid bacteria. It may be interesting that on the vegetable, lactic acid bacteria represent about 0.1% of the bacteria which are on the vegetable. So we have only a few lactic acid bacteria, and now we have to somehow promote the growth of the lactic acid bacteria. And this is done by salt. So we add 1 to 3% salt, and this gives the lactic acid bacteria an advantage. And already in the first few days, the heterofermentative lactic acid bacteria, they start to grow and they produce again, you can see here, not only lactic acid, but they produce also the CO2. And the CO2 is in the beginning very important because the CO2 replaces the air that is still present here in the headspace of your jars. So the CO2 forms then an anaerobic atmosphere above the brine, and this is very important. So therefore, first we have the work of the heterofermentative lactic acid bacteria, and after about 1 week, we have a pH then of 4, and we have a lactic acid concentration of 2% here on this fermentation stage. And then we have a shift because the heterofermentative lactic acid bacteria, they are not as acid tolerant as the homolactic lactic acid bacteria. So we have a switch, and the homofermentative lactic acid bacteria are mainly replaced then by these homofermentative lactic acid bacteria, which you can see here, the red bacteria, and these bacteria produce then even more of the lactic acid. And for sauerkraut, for example, at the end of this fermentation, after 30 days, we have a pH around 3.5 and about 2.5% lactic acid here in the fermentation. So CO2 is produced during the heterofermentative lactic acid bacterium fermentation, and therefore gas formation is a sign of an active fermentation. So you should observe gas formation, in particular at the beginning of the fermentation. Sometimes this might be delayed because the process starts not always from scratch so easy. It may take some time. Other fermentations are very active for the first few days and then settle down. So the heterofermentative bacteria are very soon replaced by the homofermentative bacteria. And as a hint, gas development should occur within the 48 hours.
Start it once more.
I think it does not work. So you have to imagine there, a lot of bubbles formed by the heterofermentative lactic acid bacteria.
That was after 48 hours. And it's the carrot you have on your plate.
Now the question is, what is if we observe no formation of CO2, no gas formation, maybe even after 3 days? What's the recommendation?
Well, I would use the vegetables as food normal. I would take them out of the brine, clean them a little bit, and then use them as food, because it won't work anymore. It's just, it didn't work out of some reason. Whatever, Susan, whatever.
Iodized salt.
Yeah, iodized salt, but it can also be sold as organic food, but it wasn't organic food or something like that. And then you have a lot of poison on top of it, which just doesn't work either. So there are a couple of reasons. Sometimes you don't know why, but yeah, there is a trick, and we'll come to that at the end.
Perfect. I mean, just what the important thing is that you do not go on with the fermentation.
Exactly.
Yeah, because very probably you have not a low pH, so it's not protected from pathogens. So therefore, take it out and eat it, and don't put it on the shelf for another 2 weeks.
It won't help. It will do others.
Okay, so I think now it's time to go to the second fermentation, which is kimchi.
Mm-hmm. Okay, so we're going to bring you through a kimchi fermentation, but please, I'm a Swiss guy. I'm not a Korean. I'm not talking of a kimchi which somebody with Korean relatives or even Korean would say, this is kimchi. We're talking of a Western-style kimchi, okay? Mild. It's the middle on your tray, a small about. First of all, we need some kind of a vegetable. Classical one is Chinese cabbage, but you can use really almost everything. I love kohlrabi. Kohlrabi kimchi is something really, really great. It's now season for that. Pumpkin kimchi is perfect, things like that. Then you need some kind of a spiciness. We use normally a classic chili, some kind of a chili, or the other one is then we have here the classic kimchi from Korea, which you can also use. There, please look at the package. Some of them are colored, so that it really looks red. That's just that it's more like what they want. Then we have our, oops, I'm a little bit ahead, sorry. We have a leek. If it's season is, or in spring, you can use spring onions. Then we take a radish, and the radish you see here is already slightly dried. We we keep our radish a couple of weeks so that it loses a little bit of moisture because we have anyhow enough liquid in our kimchi. So that's why we do that. Then you have the option of using fish sauce, or you can also use soy or the soya sauce. In our case, it's not a soya sauce; it's a pea sauce. There, if you want, then you can put a little bit of seaweed into it to give it a little more fishy flavor if you want to make a vegan kimchi. Then, for my opinion, very important to give it the real kimchi taste is fermented garlic and fermented ginger. And the ginger has also a controlling part, which gives the whole fermentation, calms it down. It doesn't treat it, but it calms down. It doesn't make it that aggressive. Then our salt, and we need something between 10 and 25%, 25 grams of salt. So that's 1% to 2.5% of salt, and I'll come to it why it's at this range. And at the end, there's the sugar. And the sugar just helps to start the fermentation and gives more acidity. So what we do is first, we cut the cabbage and sprinkle it with salt. That we do the evening before. Then you see in the morning you have this water, this liquid from the cabbage, which has gone lost over osmosis. It's left. And then what we do is we rinse it, clean away the salt, and after that we chop it down. We peel the garlic, and my personal favorite to peel garlic is a spoon. Works perfectly. Just scratch off this brown skin. Did I say garlic? Sorry, ginger. Thank you. Okay. I said ginger, not ingwer. I haven't said it yet. Okay. Then we put everything in our molder. Well, this is for personal. We have other things to make when we make kimchi, we make about 100 kilos.
That's for a nice evening. That's for one evening.
We add sugar and salt, and then we grind it down. Chop the onions. And we have this mix. In the bottom, you see the cabbage here. Here's the cabbage. Then we have the onions, and on top we have this ingwer. Bravo, ginger.
There you go.
Ginger. Now you can clap. Okay. We have the ginger and the garlic together with the sugar and the salt. Then chop up the chili, and I would use a glove because that can really hurt if you put it in your eyes. That's why I have a glove. Then we knead it down very well. Really, you can use energy for this. And then you see here how the moisture comes out of the product. There's a lot of water here also. And you'll see it when we pack it up, how much liquid we have. And here you see it, the brine, as Markus said, under the brine is fine. You see here's the brine edge, and the kimchi is under that. That's the whole story.
Thank you, Patrick.
You're welcome.
So this is the delicious part. Now I want to talk about food safety aspects a little bit. Oops, there's some more kimchi. Okay.
The radishes, Where did that go? Okay.
Okay. But now, food safety aspects of fermentations. And I have to state at the beginning that lactic acid fermentation of vegetables is one of the safest fermentations that you can do. So we know much more complex fermentations. For example, if you ferment fish or if you ferment meat, then this is a very complex and also very dangerous fermentation. So you should be really experienced if you ferment fish or meat products. But even for the fermentation of vegetables, there is some food safety aspects. And one problem is that we have contaminated raw materials. So the main concern with fermentation, vegetable fermentation, is from contamination of raw materials and also fermentation failure. So this is the two major aspects that we have to keep in mind. The contamination very often occurs already in the raw materials that we use for the fermentation. It can also happen here during the production process, sometimes also after the fermentation at the distribution stage. That would also may happen. And then we have the problem of a fermentation failure. And we already discussed some reasons for such a failure, for example, the iodized salt, or maybe also insufficient salt, so other bacteria have still the possibility to grow and overgrow then the lactic acid bacteria. And there's even another reason. You can see it very well here on my shirt. It's bacteriophage. Bacteriophage is viruses that infect bacteria and kill the bacteria. This is very often a problem in dairy fermentation, and it could also happen here in the fermentation of vegetables. So what's very important is that fermentation does not replace general food safety principles or food hygiene. And that means the raw materials that you use should always be the best raw materials, in the best case organic raw materials, and not the rotten ones that you won't eat without fermentation. Yeah? And also kitchen hygiene is very important, in particular when you're setting up or when you're preparing then the fermentation. This is the three major health concerns in vegetable fermentations. So pathogenic bacteria, as already mentioned. Then mycotoxin-producing molds. This is really a topic in vegetable fermentation also. And then these biogenic amines here. Read these names, putrescin, cadaverin. So I think even from the names, you get an impression how they smell. And this is true. Sometimes they are also called smell of death. And this is the reason because these molecules are formed in high concentrations on decaying animal bodies, dead bodies, for example. Yeah? So this is a typical smell of dead bodies. And then we have histamine, for example, also belongs also to this biogenic amines. And the problem with the biogenic amines is that they may produce symptoms similar to food allergy reaction. So rash is very often a symptom. Flushing, itching, and also digestive issues can be caused by these amines here. The biogenic amines are produced from amino acid by decarboxylation, and that's the reason why they are in particular relevant in protein-rich fermented food. So again, we have fish fermentation, meat fermentation, where these biogenic amines play really an important role, but also dairy products like long-ripened cheese, for example, is also often containing high concentrations of these biogenic amines. This is not the case in vegetable fermentation, and therefore I will not focus more into detail on the biogenic amines. I just want to switch to the pathogenic bacteria and to another problem that we have with the pathogenic bacteria in vegetable fermentation. And I already told you that they may already contained by the raw materials. And the problem that we have in vegetable fermentation is that we have no step where we inactivate the bacteria and pathogens. Yeah? So we have no heating step, no pasteurization step, like for example for milk products that you ferment. So if you have alive pathogenic bacteria in your raw materials, they will also end up in your fermentation. So this is a list that shows some of the pathogens that may be contained by your vegetable fermentations. And I highlighted Clostridium botulinum because Clostridium botulinum is one of the most dangerous foodborne pathogens that we have. The bacteria are ubiquitously around in the environment, so they are everywhere, and they are also on the vegetables that you use for the fermentations. And these bacteria, they like anaerobic conditions. So under anaerobic conditions, conditions, they grow happily, and during growth, they produce then the deadly botulinum toxin. So therefore, not only Clostridium botulinum, but also Clostridium botulinum growth has to be controlled. And as we already said, this is done by the pH. And a pH of 4.5 is kind of a magic pH in food microbiology, because at a pH of 4.5 and lower, there is no growth of pathogens, no growth of toxin-forming bacteria, and also most of the spoiling microorganisms are also inhibited at a pH of 4.5. And as I told you before, when we talked about the steps in the fermentation, you heard that very often we have, after a few days, already pH 4, or we have even a lower pH than 4. So the pH is very important, but, oops.
Oh, yes, sorry, I couldn't resist. I couldn't resist adding an equation. Yeah, exactly. So pH is very dangerous because it looks like this. It's the minus logarithm of the concentration of hydrogen ions. And so logarithm means also that if actually one step in pH means actually a 10-fold change of the concentration of these ions. And that's all I wanted to say.
Fine, but much more important is that it works. So this is really crucial, the low pH, and together with, we already discussed that before, the interaction with other bacteria. So it's not only the low pH in the fermentation, it's also the competitive microflora of lactic acid bacteria that compete with other bacteria, with unwanted bacteria, pathogens within this fermented food. And then we have a second very important health aspect, and this is the mycotoxins. And the adverse health effect of mycotoxins is one is the immediate or the acute poisoning that can happen, and then we have some long-term effects, which might be, for example, immune deficiency and even cancer that can occur due to the ingestion of these mycotoxins. Therefore, it's really important that we avoid any intake of food that may contain mycotoxins. That's really important. And therefore, we have to look on our fermentations and to try to identify whether we have mold growth on the fermentations. This is very easy here. And this is also how it typically looks like. So if you have mold growth, you usually see this hairy or this felted surface. And this is sometimes green, sometimes blue, gray. Other black. Colors are also black. Also other colors are possible. And what is important to know that mold growth only occurs in the presence of oxygen. And this tells us why very often we have this situation. So you can see that this part of the vegetable is not under the brine. It's above the brine level, and therefore there must have been also oxygen in the headspace, and therefore the mold could grow here. And this is what Patrick also already mentioned. One of the golden rules in the lactic acid bacteria fermentation is if it's under the brine, it's fine. Because under the brine, there would be no growth of mold and no formation of mycotoxins. However, this would not mean that you can take out now this moldy part and eat the rest. Because what you see here is the fruiting bodies of the mold, but the mycelium penetrates deep into the tissue of the vegetables, and the mycotoxins, they are secreted, so they diffuse into the brine. So everywhere in the brine, everywhere in the fermentation, you would have these mycotoxins. And therefore, another important thing is if you identify mold in your fermentation, you have to throw out really the whole fermentation to be on the safe side. That's important.
If you imagine, here in this case, if you imagine the only thing you see with the mold is you see the apples, you don't see the tree. And that's why you have to throw everything away.
Do you want to see one example of the mold?
We can try.
Yeah.
It depends on how good the cameraman is.
Yeah, cameraman.
Okay, and we have one at the side. This one.
I mean, you can clearly see the hairy surface. Yeah? So that's really an indication for mold.
And the even nicer one, we're going to try to pick that one out, is here. Not.
The black bit.
It's there, yeah.
There it is. Don't move, Patrick. That doesn't work. Let me, so. What?
Excellent. So I think now it's time to go to, oh, there you go, nice. Yeah. Oh, that's disgusting. I think we should do something a little bit more delicious. What do you think?
So very important is also don't smell on this mold. Yeah? It's also dangerous.
So we have our third fermentation, which is the lacto water.
Yes, next fermentation. Okay, so what we're now going to do is I'm going to show you a very, as always, a very simple fermentation. And with something which I personally think is something that those of you who want to try to make some fermentation, things like that, really, it's a big help. What we're going to make is a wet brine fermentation with carrots. That's the carrot you haven't ate yet. That's that. I've already prepared, well, not me, but one of my employees have prepared the carrots. She told me I can put them straight in, so I'm going to try to put them straight in. You see, I haven't washed my hands now for this process. I would have to go out now, but in a kitchen, I would suggest just wash your hands and then you can start to ferment. It's no problem at all.
So the fermentation will start earlier.
Huh? This one's going to start pretty well with my hands, yep. And I had my hands the whole day in sausages, so they're pretty clean. So, okay, so I've packed the carrots. Then I put about the same amount of water to it, which is here about 500 grams. Julia already prepared the water, so it's very easy in this case. And let's see that Markus is happy with me also. Under the brine is fine. Okay, and then I put 7.5, right? Yep, 7.5% of, not 7.5 grams of sugar. That's to start the fermentation. You can use, by the way, you can also use Birnel, which is a syrup, exactly, thank you. And then salt, I'm going to take 3%, which means I need 15 grams on this one. And now, look away, or you can now look. This is what we call Lacto-Wasser, lacto water. It's from the carrots you have eaten, the water, and we use that always to donate some of the bacteria into the brine, which gives us a very fast starter. So we're not waiting and hoping we'll get a start. Here we already have it, and you can use it for almost everything. I wouldn't take celery or something like that because that has an allergen in it. I would take carrots or something which is safe, and you can use it in sweets, in strawberries and whatever. It doesn't really matter, and you just put really a small amount. It's just that's it. That's the whole thing. Now, what you can also do to give it a little bit of safety is you can peel off a little bit of ginger. Yeah. Thank you. I don't have any formulas, so that's why I don't get any clutches. I'm going the whole work, and then So take 1 or 2 of these, put them in, seal the lid. That's it. Put the date on it. 3 or 4 days later, you'll have your fermented stuff. Go ahead.
Fantastic. And also now we thought we would actually show you a little bit under the microscope what this water here looks like. So you can actually see some of the bacteria. So this is how it looks like. So Markus, can you describe us a little bit what we see? I hope you recognize some of the people here in the picture.
So as what you see is as we promised before. So it's more or less 2 different morphologies. It's a single cocci, like this one. And then we have straight rods, the bacilli, which is this one. And then we have some that look like rods, like this one.
Or this one, yeah.
Or this one. But if you have a detailed look, you see that is cocci forming a chain. So we have these different types, the rods, and these cocci, streptococci, and also these single bacteria here. And I mean, this is a small drop of this lacto water. And in this small drop, we have approximately the equivalent of the world population of humans. So therefore, we don't need that many. So it was just a few milliliters maybe that Patrick added. So it's a huge amount of bacteria, of the bacteria that we want, the mixture of heterofermentative and homofermentative lactic acid bacteria that was added to this fermentation.
Perfect. And then I think now we have our last part, which are frequently asked questions. So we thought we would summarize some aspects that, particularly from you 2 guys that have experience with this, have come up often as questions. And so we're going to try to break them down here at the end.
There's one more thing you have on your plate, and that is a salt lemon, which is also a lactic acid fermentation with a lot higher salt condition. So try that one also. Okay. We have a couple of slides to a couple of the frequent asked questions, but first I would like to hear from you. Is there anything you want to know? Yep.
Is any mold so dangerous, like mold on cheese?
That's a question for you.
Mold on cheese is dangerous if it's not a mold-ripened cheese. So if it's camembert or if it's a blue vein cheese, then it's fine, of course. But if you have mold on cheese that should not be there, then you have to be careful. Because then, no, I have to say, if you have a hard cheese, if you have a very long ripened hard cheese, then this would make sense if you have one tiny place where the mold is, so you can cut this away. But not on soft cheese. And if you have everywhere these parts of mold, then throw it away to be on the safe side. This is always the question: how much do I have to take away? So therefore, for being on the safe side, throw it away. So maybe we start.
Should I add some vinegar to my wine to maybe save it first?
No, if you use vinegar, then you're pickling. You're not fermenting anymore. No. Stop it. It's not the same. It's just not the same. You don't get this beautiful acidity if you pickle something. That's completely something else.
Are there some famous spices that we should avoid always when we're fermenting things?
Spices?
Can you add pepper, cumin, whatever?
Just the most important thing is good quality. Really good quality. I think spices is a big problem because to get really good quality is the main issue. And then you can almost use any spice. There's not, I don't.
You're right, but I wanted to mention that I think we should go further. Maybe we answer already some questions that would be in the auditorium, and then we have again time at the end for questions and answers.
Okay.
And my first question would be, would you eat this? Who would eat this? One person, 2, 3, around five people from 320. So, to be honest, it looks really disgusting, but it's not mold growth. So sometimes it's difficult to distinguish between mold growth and this growth. And what this is yeast. So we have the growth of yeast on this on the surface, very often on the brine, on the brine surface, and this is the so-called calm yeast. So calm yeast is usually flat, thin, white to cream-colored powder. It grows when it grows thick, it looks like velvet, but it has not this hairy surface structure than mold. So sometimes then it's difficult, but if you are not sure, you should be on the safe side.
Yes, and we can actually show one example.
Yeah.
Yeah. I think it looked better in your picture, Markus.
But you can see it's not hairy. You see sometimes it's like the surface of some cheese also. It have this surface, and if you want, you can come in front later and have a close look, and you will see it's not hairy. So we can really eat this. We can remove also the calm yeast. That's also not a problem, and you can eat it.
You can also come forward for a tasting if you want.
By the way, it smells great. It's no problem at all. It's a great kimchi.
It's from a former student. So she hasn't been sure about that and said, oh, maybe I want to have it for demonstration purposes. So when do these calm yeast occur? Patrick said the biggest problem is oxygen, again in the headspace. So if you have oxygen in the headspace, then very often this calm yeast appears. Also, if the fermentation temperature is too high, maybe above 25 degrees, and if the brine is low in salt. So this might be reasons why these calm yeasts occur, but these calm yeasts occur very often. I remember my first fermentations, I always had these calm yeast, and yeah, I didn't eat it. It looked so disgusting, but now I know it would have been possible to eat it. Okay, the very important thing is don't confuse calm yeast with mold. Yeah? So if it's mold, you should be sure that it's not mold. Only then you really then consume this fermentation product. Okay, another observation that is often made, in particular if you ferment beetroot, is that you have a very slimy brine. Very slimy brine. Somebody observed this before with beetroot? Okay, the experts in the first row. Yeah, and what happens here is that in particular the heterofermented lactic acid bacteria Leuconostoc, they produce so-called dextrane, which is very long polysaccharides, and this dextrane is only produced if sucrose is present. So dextrane can only be produced from sucrose, and look on this table here, and you can see that in particular beetroot consists mainly of sucrose. So carrots also, I don't know whether carrot also show this thing sometimes, but it's very prominent for the beetroot, and the explanation is the high concentration of sucrose in the beetroot. It's harmless, so you can still use, for example, the vegetable. You can eat it, the vegetables, the fermented vegetables. You can also put it into cooked dishes if you want, and blend it. Okay, another observation is that the tissue of the vegetables becomes very soft. This is also a very common observation. And now we come back again. So you can see here the pectin, and what did Thomas say before? Pectinases, so this is enzymes that degrade the pectin. And the pectin is part of the middle lamella, which is part of the plant cell wall, and this leads then to the softening of the plant cell walls and to the softening of the tissue of the plants. Also here, it's harmless, so you can really eat these soft vegetables. You can also use them for cooked dishes or again blend them. And then we have sometimes a smell of sulfur, and I don't know whether you observed.
Did you get the smell? A little bit. It wasn't that bad.
So we thought it's more intensive.
Yeah, I hope we would smell it here also. But
But this is quite normal, and this occurs with certain vegetables, and this is in particular vegetables that are rich in sulfur compounds, and this is the cruciferous vegetables like cabbage, cauliflower, broccoli, cauliflower. That was what Patrick brought, and what had his name. And also the allium vegetables like garlic, onions, leeks, if you ferment them, they also have this strong sulfur smell. And this is from the organosulfur compounds that are converted by the bacteria to hydrogen sulfide. And this hydrogen sulfide is a noxious smelling gas. And if you open the jar and it's too much of this sulfur smell, then you can just wait. So let it open or take it out, put it on a plate and wait some time until the smell has disappeared a little bit, and then you can eat these fermented products. And the last thing that we have, we already discussed about putrescin and cadaverin, so about these amines that are sometimes present. And if you have a lacto-fermentation, that smells really of putrefaction. Like rotten food, and if the fermentation smells, throw it out. So this is really important. So this is also not disappearing after aeration, so it stays, this smell, and if this occurs, you have to throw it out. And this is the even more important golden rule: when in doubt, throw it out. So be on the safe side. If you are not sure, don't taste. Don't taste to determine whether the food was safe. Although this will work sometimes, but maybe only once.
Thank you, Markus. Good. Any other questions? Reto.
Temperature. Can I do it in the cellar too?
Okay. Well, we have a rule. Professional fermentation is first 2-3 days we keep it at about room temperature, and then we put it at about 15 to 17 degrees, because it gives you more aroma, more taste. The slower the fermentation goes, the more taste you get. Maya. Miso. No, because that would be a topic for itself. But if somebody's interested, on Sunday, I have a course on miso at the Pure. So if somebody, good. Any other questions? Yeah, okay. Mm-hmm.
Sugar. Is there any difference in terms of, processed sugar, raw sugar?
You can use any kind of a sugar. It really doesn't matter. The less processed, the better. Really, because the point is, we give a little bit of sugar to it just to give it a good start. It doesn't need a lot. It really doesn't need a lot. It's absolutely not. And if you put too much sugar into it, you get more acidity. So it's directly converted into acidity. Hey guys, help me. I don't know whom. Okay.
Say the pH is not the same as the lactic acid amount we have in the fermentation. So meaning that in order for the pH to go to 12.6, we need much more lactic acid. Are there any vegetables that particularly we shouldn't ferment?
Oh, now I got it. Personally, what I really think is not something for to do at home is mushrooms. No wild mushrooms, and I wouldn't even say for you guys, except you're planning to give me a concurrence, but then I would say no mushrooms, because they don't have really that much sugar, and you really have to be really aware of what you're doing there. That's my personal opinion. I couldn't say this one or this like that.
I think we have time for maybe 2 more.
We haven't do anything about means of therapeutics, maybe where fermentation can have an impact, maybe for medicine?
I hate this question. No, because they're like two groups. What I like to talk about is prebiotics. That means what we do is we do a digestion in a glass, mainly with things which we can't really digest that well, like cabbage, like dairy stuff and beans. And so we do that digestion in a glass, and then it's good for us. It's easier for us to digest it. That's the part I think is personal, my opinion. And about probiotics, I don't want to talk because, yeah.
Just to this question, there is, from the scientific viewpoint, the question is easy to answer. It's a no. Because only yogurt so far is accepted as really a health contributing fermentation product. All other products are in discussion, but there is no scientific proof, no clinical study that shows that certain fermentation products or also certain probiotic bacteria have really a health advantage in the human body.
I think we can take the last question. Maybe from this side.
Please, we'll take both.
That's the question about calm yeast. Is this related to what we call Essigmutter?
Nein.
It's not.
No. It's yeast. It's a yeast. But it's a yeast which really doesn't do much to the product.
Vacuum bag fermentation, blew up the bag after roughly 1 week. And then I punched a hole in there to let it pressure and resealed the bag, and then it didn't do anything.
Yeah, sure. That's what. Did you listen this evening? Can you go back to the slide? No, we have two kinds of bacteria types. And the first ones which start are the heterofermentative, and they produce CO2. And the second group, which stays longer, they don't produce any CO2 anymore. So that's it's perfect what you said. So thank you very much, and it was great to see you here.